Hello everyone, welcome back. We're going to be going over photosynthesis now. I hope all of you were able to review cellular respiration and also review your guys' lecture material for photosynthesis. I'm not going to go into as much detail as I did with cellular respiration. So I'm just going to give a quick introduction to photosynthesis. Hopefully you guys have both read your textbook and watched the lecture your professor put up for photosynthesis. So essentially, so now we're just kind of seeing that cycling of cellular respiration and photosynthesis. You're going to see that cycling happening. Now that you understand cellular respiration, this should really be a piece of cake for you because as you can see, our end products here are going to be essentially what we need for cellular respiration. So our reactants will be carbon dioxide and water. We'll use light energy in the form of sunlight to generate glucose, energy, and oxygen. And we typically think about plants undergoing photosynthesis, but there are certain bacteria that can undergo photosynthesis. Algae can do this as well as protists. So, but I'm just going to talk about it in the context of plants. So the point of photosynthesis is essentially to capture sunlight and make energy. We're trying to make glucose from it because remember, we can cash in this glucose in cellular respiration for a ton of energy and oxygen. Okay, so here is an overview of photosynthesis. So photosynthesis is going to occur in two phases. So this over here is what we refer to as the light phase. So this is going to require light. It is dependent on light. This over here is what we typically refer to as the light independent phase or the Calvin cycle. I usually call it the Calvin cycle. You will see older textbooks and older material will refer to this as the dark phase. But that isn't necessarily true. This phase does not require darkness to work. It just doesn't depend on light. So it is a light independent phase or a Calvin cycle phase. I will accept both answers. So we have two phases. We have our light dependent phase and our Calvin cycle phase. And both of these are going to occur in the chloroplast. So this structure shown here is the chloroplast. So if you remember back to our previous instructions or our previous activities, we saw chloroplasts in the Elodia plant in the microscope. They were those green oval structures. So that is where photosynthesis is going to occur. And as you can see, we have water, carbon dioxide, and light energy coming in. So we look back over here, we have carbon dioxide, water, and light energy. So carbon dioxide comes into the Calvin cycle, water comes into this light dependent phase, and then light, sunlight. And then what are you going to get at the end? You're going to get oxygen and you're going to get sugar. So it's shown here as sucrose, but you can also get glucose. So we tend to think of it with glucose. So the light phase occurs first, and it is shown here. This takes place in thylakoids. So these are disc-shaped structures that are found inside the chloroplast. They're not shown in this image here, but they are shown in a further down the line. I'll show you guys the structures. And these thylakoids have this thylakoid space and these thylakoid membranes. So they kind of have that same setup that we saw in the mitochondria for cellular respiration. The light phase requires light, hence its name, and it results in the production of oxygen. The second phase is what we call the Calvin cycle phase. Oh, I'm sorry. And in the light phase, you're going to generate ATP and NDPH. And this is what's going to fuel or give the energy needed for the Calvin cycle. So this is what's going to get the Calvin cycle going. In the second phase, the Calvin cycle, it takes place in the stroma. So it's going to take place kind of in that fluid-filled area of the chloroplast. And the CO2 is going to come in and be built into a this right here, which is a 5-carbon molecule. So the CO2 will come in, interact with this 5-carbon molecule to make we'll go into it, but eventually it'll go around and make glucose. Remember, we need ATP and NDPH to get this going, and we also need carbon dioxide to get the Calvin cycle going. Okay, so let's get into it a little bit better. So now we're going to discuss the light reaction phase. So we're going to discuss this a little bit more detail. So kind of think of this phase like the electron transport chain in cellular respiration. So since we learned that concept pretty well, this photosynthesis is going to be extremely easy. So what happens here is essentially you're going to have these electrons that are moved along these photosystems here, and they're basically moved along these proteins, these molecules, etc. And as these electrons are moved along, you're going to have protons that are going to be pumped. So here the protons are going in the opposite direction than what we previously saw. So here the protons are going from the stroma into the thylakoid space. And as these protons are being pumped in here, they're going to create that concentration gradient. And then these protons will be able to come over here, interact with this ATP synthase, which we already discussed. When the protons come to this ATP synthase, it's able to create that rotation in the ATP synthase. Remember that gamma subunit, and then the beta subunit, which is this head, is able to, in the loose configuration, it's able to have ADP and a phosphate bind. And this is the loose configuration. In the T configuration, the ADP and the phosphate come in close proximity. ATP is formed. In the O, or open configuration, ATP is released. And remember, we're going to need this ATP in the Calvin cycle. Okay, so that's very briefly. So the process begins in photosystem 2. So you might be asking, why is this called photosystem 2 if it's going to begin here? It's just essentially photosystem 1 was identified first by scientists, so it was called photosystem 1, and photosystem 2 was found after. So everything begins here at this photosystem 2. And then a photon of light will come in. So you see this photon of light will come in and get this whole process going. So you don't really need to know this, but I'm still going to tell you. Photosystem 1 and photosystem 2 require different wavelengths of photons in order to absorb the light. So you have a photon of light coming here, getting this system going, a photon of light coming here, getting this system going. They both require different wavelengths to get their systems going. So the photon of light is going to hit photosystem 2, and there's going to be chlorophyll in here. This chlorophyll has electrons that can be excited, and they're going to be able to leave the photosystem 2 structure. These excited electrons will pass through a series of electron transport chains that are embedded in the thylakoid membrane. So they can pass these structures, the electron transport chain. And ultimately, these electrons will end up at the photosystem 1. So they end up here. So just follow this orange arrow, I guess, for now. As the electrons move through the system, remember, protons are going to be getting pumped from the stroma to the thylakoid system. Into the thylakoid space. And then, let's see, is there anything I missed here? Okay. So here, the protons will come interact with this ATP synthase, and it will allow protons to go through and make ATP, which we have already discussed extensively. And after the electrons go through the photosystem 1, so we're still here with our electrons, as they go through the photosystem 1, they you will see that you end up with NADPH. So these are electron transport carriers, and this NADPH is going to be used in the Calvin cycle. This ATP will be used in the Calvin cycle. Both ATP and NADPH are essential for the next phase, the Calvin cycle, because they will need this energy source to pull that carbon dioxide out of the atmosphere and build it into glucose. So that's essentially what's going on here. So I just want to backtrack to photosystem 2 real quick. So remember I said that the photon of light is going to come in and hit photosystem 2. And chlorophyll has electrons that can be excited, and they're able to leave photosystem 2, go through this electron transport chain into photosystem 1. Well, what do you think is going to happen when the electrons from photosystem 2 leave? So when these electrons left to go do their thing over here, they left holes in photosystem 2. So these holes essentially have to be filled. So it's not shown in this image here, but essentially photosystem 2 is going to pull 2 electrons from the manganese complex to fill its holes. But now guess what? The manganese complex has 2 holes, so it will pull 2 electrons from water to fill its 2 holes. So it's going to pull 2 electrons from water, and then you're going to be left with this half of an oxygen or an oxygen. So what happened is that we essentially used light, we used water, and we ended up with oxygen, we ended up with ATP, and we ended up with NADPH. These are going to be our energy sources for the next reaction, the Calvin cycle. Remember that the light reaction phase is going to happen here in the thylakoid structures, and the Calvin cycle happens here in the stroma of a chloroplast. Okay. Okay, so now we move on to the Calvin cycle. So what's happening here is, well, first this takes place in the stroma of the chloroplast, and essentially CO2 is taken from the atmosphere and it's going to be made into a molecule. So like I said, this requires energy, and this is where our ATP and our NADPH are going to come in handy. So this molecule will go through essentially many modifications where it can be made into glucose. So it's going to be essentially this G3P molecule. And if you remember from my glycolysis reactions, we had glucose that came in as a 6-carbon molecule, and it broke up into 2 3-carbon molecules called G3P. Remember, it originally broke up to 2 3-carbon molecules, the DHAP and the G3P, but that DHAP underwent some modifications, became G3P, and we essentially had to follow just the same 3-carbon molecule down glycolysis. Okay, so CO2 is going to come in, interact with this 5-carbon structure that's already there, going to go undergo many modifications. What's going to fuel these modifications are ATP and NADPH, and you will eventually have basically glucose formed, or it could go back into the structure into the cycle where it will become that 5-carbon molecule, interact with CO2 again, and the cycle will continue. So I just want to go through this with a little bit more detail. But essentially here you have, this is your simple image. This is your, I guess, more complex image. It's still not very complex though, so don't get worried. You essentially have 3 phases in the Calvin cycle. First phase is what is referred to as carbon fixation. So here you have your carbon dioxide come in, and it's going to interact with this 5-carbon molecule that you have already there. And this 5-carbon molecule is ribulose bisphosphate. Don't need to know that. Just know 5-carbon molecule going to interact with carbon dioxide. It's going to form this 6-carbon molecule, but this 6-carbon molecule is super, super unstable. So it's going to basically immediately break into 2 3-carbon molecules. So it breaks into 2 3-carbon molecules here. And then you're going to have reduction occur in phase 2. So phase 2 is reduction. So I briefly mentioned in our cellular respiration lecture that oxidation is lose an electron, oxidation. Gain an electron, reduction. So in phase 2, we have reduction occurring. And here, these structures, these 2 3-carbon structures, are essentially going to gain a phosphate from the ATP. So ATP is going to leave as ADP, and these are going to gain phosphates. And then this phosphate is then going to get reduced by NADPH. So it'll get reduced by NADPH, and then you're left with this G3P molecule. So G3P is an energy-rich sugar molecule that can be used to make glucose as well as other organic molecules, such as cellulose and starch. So it could, oh, it shows it here. So it could be made into cellulose and starch, which are used for long-term or storage, or it can be used for glucose, which is, you know, that short-term energy that our bodies like to consume. Okay, so now we're going to move on to activity number 2. So I told you guys our photosynthesis lecture was going to be much shorter this time since your guys' videos are up. I hope you guys were able to watch those. Your photosynthesis videos from your lecture portion are up. Let me rephrase that. So I hope you guys were able to watch those. So now we're going to move on to activity number 2. So I would first like you to read the setup in your lab manual before we go over this, but you should always be reading your lab manual before you watch these lectures. So essentially here, we're going to be measuring CO2 use by Elodia. Remember, Elodia is this freshwater plant that we use. We've used it before in our lab, and we're going to use it again today. And so let's think about this. So we're measuring CO2. Which phase of photosynthesis do you think we're measuring? Let's go back. So we're measuring CO2 utilization. So what phase do you think we're going to be looking at here? So yeah, correct. We're going to be looking at the Calvin cycle, or the light independent phase. Okay. So here we're going to measure CO2 utilization, or the amount of CO2 consumed. And during this Calvin cycle, plants use CO2 from the atmosphere and energy molecules produced during the light reactions to produce glucose. So here we're going to use this indicator, phenol red, to measure whether CO2 utilization is occurring. And let me go ahead and introduce this phenol red dye real quick. So this is a pH sensitive dye, and essentially it can measure whether a solution is basic, neutral, or acidic. And the way it does that is when the solution is basic, so it has a basic pH, something like pH of 8, you will see that this phenol red dye is going to have this red color. When the pH becomes something more neutral, like a pH of 7, the dye will change to a more orange-like color. And then as it becomes slightly acidic, so it has a pH of something like 6, the dye is going to become yellow. So that's essentially how this phenol red indicator is used in labs, and that's what we're going to use to visualize this process today. Okay, so you're going to grab a beaker, you're going to fill it up with your solution, and you're going to add some phenol red to your solution. At a basic pH, your indicator will be red, but at a low pH, the indicator is yellow. So the first thing we have to do is we have to decrease the pH of the solution. So the way we're going to do that is we're going to put a straw in our solution. So here we go. We're going to put a straw in the solution and we're going to blow air into the phenyl red solution. And what we're essentially doing when we're blowing and we're creating bubbles, so we're going to use the straw to create bubbles into the solution. And while we're doing that, we're introducing CO2, carbon dioxide, into the solution. So as you can see here, you have water and carbon dioxide here. And as these two molecules interact, they're going to form a weak acid, which is called carbonic acid. So as you blow your CO2 into the solution through the straw, the solution will turn from a basic red color to a more yellow color because remember, we're forming this weak acid, which is carbonic acid. Okay. So next we're gonna, so now we've already gotten our solution to this acidic pH. We're gonna put 10 mils of our acidic solution into 3 test tubes. Our first test tube is going to be our control group. This control group will have no plant in it. Our other 2 experimental groups will be group B and group C. Both group B and group C will have this freshwater plant in it, so the Elodia will be placed in these test tubes. Now the difference between B and C is that B is going to be a other form of control group. So well, I wouldn't consider it a control group. It's essentially just going to have no light. So this group B or condition B will be covered with foil so that it does not have access to light. And then group C will have the Elodia plant in it, which can undergo photosynthesis, and then it will also have access to light. Remember, light is one of those things that we need in order for photosynthesis to occur. So all test tubes are going to be monitored every 15 minutes for a total of 2 hours. And essentially, as the solution becomes more basic with the uptake of carbon dioxide by the Elodia, the solution will also become more red. So if the Elodia is using the CO2 in photosynthesis, then the solution will become more red or more basic. So you're going to essentially monitor the color of the solution for 2 hours. So pretend 2 hours passed. Oh, here's some things for you guys to do independently. So after 2 hours passed, you're going to look at your results. Your control group, you will see, maintain this acidic color. There was nothing in there to influence this condition, so it remained the same. Here we put the Elodia plant, but it didn't have access to all the components a plant needs to undergo photosynthesis. So you want to look at the solution around the plant. You know, it's kind of difficult to see, but you want to look at the solution. You'll see it's like a yellow-orange color. Okay, it's acidic neutral. And then you want to look at condition C. And you will see that it is a red color. It has a basic pH. So that means that this Elodia plant was able to utilize this carbon dioxide for photosynthesis. So then I'm just going to have you guys think about this a little bit. So how do you think you could have sped up the process of photosynthesis or turning the solution red? I want you guys to draw some conclusions. And that's about it for activity 2. So now we're going to move on to activity 3. So for activity 3, we're going to be measuring oxygen production in Elodia plants. So once again, we're using this freshwater plant Elodia. And here we're going to be measuring oxygen production. So let's think back to photosynthesis. Remember, it has 2 phases. Where is oxygen produced? Is it produced in the Calvin cycle or is it produced in the light-dependent phase? Okay, correct. It's produced in the light-dependent phase. So also shown here, we produce that oxygen. Remember when the water is split to replace the electrons that were lost from that photosystem too, we create this oxygen here. Okay. So here you're going to be measuring the amount of oxygen produced by the Elodia during during the light reactions. So here you're going to this is going to be your experimental setup, and I actually have 2 videos attached. They're very brief videos, about 5 minutes or less each video, and they conduct this experiment under different parameters, but it's still a really good visual to see. So here you're going to have a beaker. So these glasswares are called beakers. This is a 250 ml beaker, but the book instructs you to use a 1000 ml beaker. So pretend this is bigger. So we're going to be using a 1000 ml beaker, and you're going to add 400 mils of sodium bicarbonate solution, and then you're going to fill the rest of the beaker with tap water. So the sodium bicarbonate solution is a good source of carbon dioxide. And remember, we have to use carbon dioxide in the Calvin cycle. That's what's going to interact with our 5-carbon molecule to make that 6-carbon molecule that then immediately breaks into 2 3-carbon molecules. We're going to add a few sprigs of this Elodia plant, and then you're going to cover those sprigs with a funnel. So you want the funnel to be inverted. You want the bottom of the funnel covering the plants, and then you want this part sticking up, the narrow piece sticking up. And then the narrow stem of the funnel should point up, but still be underwater. You're going to fill a large test tube with water. So right here, this test tube will be filled with water, and it will cover the narrow stem of the funnel. And then you basically want air in this test tube. Okay, I'll let you guys read this setup in your book. And then if we were in class, I would have one class leave their experimental setup, so this setup, just on their desk, so it has access to some light. We would have another setup outside where it has access to a good amount of daylight. And then we would put another setup, so I instruct a group to put their setup in the cabinet so that it has access to no light. So we have no light, some light, and then daylight or natural light. And then you're going to measure oxygen production. So basically what you're going to see here is that, oxygen will be produced up here and in these forms of bubbles. And as the oxygen is produced, this water is going to decrease. So think about this. Which condition do you think would lead to the most production of oxygen or these bubbles? So you might be thinking the condition that has access to natural sunlight, and if so, you are correct. So what you will see is when you have access to natural sunlight, there will be more bubbles up here, and this water will be displaced. It will decrease. And then the condition that has access to no light, you wouldn't see bubbles or the water being displaced. So these are the 2 videos I would like you guys to watch. They show how this experiment is conducted, and they show you their results from this experiment. And then I want you to also answer these questions in your lab manual on your own. This is for you to start thinking critically about these experiments and these concepts that you're learning. And then activity 4, I'm going to let you guys do this on your own as well. So here you have your 2 stages of photosynthesis. You have your light-dependent reactions, and you have your Calvin cycle. And you have to determine what were the starting molecules for the light reaction. What were your end products? So remember, if we go back to the light reaction, what were our starting molecules? Our starting molecule was light. Our starting molecule was water. And what was our end product? We had oxygen that was generated. And then we also had NADPH and ATP that were generated to be used in the Calvin cycle. So now going into the Calvin cycle, what were our starting molecules here. We had our CO2 that came in. Remember, it interacts with that 5-carbon molecule over here. We had ATP, and we had NADPH, which is also shown here. Our carbon dioxide, our water, our light energy, and then remember we also have that NADPH and ATP. And then what is generated? Essentially, let's see here. So we have glucose generated because remember the oxygen is generated in our light-dependent phase. So our glucose molecule will be generated here in the Calvin cycle. So go ahead and fill that out in that chart for activity 4. Yeah. And then if you guys have any questions, please let me know. You guys do have worksheet 5 to complete. I believe worksheet 5, yeah, worksheet 5 has a graph that's also included. I'm not going to have you guys do the graph just because we're not generating data and I don't actually have any data for you guys to plot. So I just want you guys to answer those questions and then send that back to me by Sunday, 11:59 p.m. And you will also have a quiz 5 to take by Sunday, 11:59 p.m. Okay, thank you.